So now we start the scientific session. What I wanted to say to the speaker is that they should explain in a simple word for the young people there. And also, the first question will come from you. So the first speaker this morning is Ravram Eshkok, who is yes, he is. And he will talk about the ubiquitin system for protein degradation.
Well, I'm very glad to participate in this conference of Science Without Borders. And I agree with the previous speakers. Science has no borders. It's international. It's universal. So we are very glad, we Israeli scientists are very glad to share our science with our colleagues from France and from all over the world. But as you have heard, this is also a meeting on celebration of the 70th birthday of Israel. So maybe it is appropriate to tell you about some of the advances, some of the contributions to international science that have been made by Israeli scientists during that time. So about my work, I am working on the problem of how proteins are degraded in cells. I'm working for many years on that. And maybe I should tell you, those who are not in the field, what are proteins? So proteins are the machines in our body. They carry out all the different processes in our body. They carry out all the different chemical processes. We have thousands of different chemical processes that we need to live. And these are carried out by proteins called enzymes. Each enzyme promotes one certain chemical process. And proteins also carry out all the physical processes in our cells, like the contraction of our muscle, like the electric activity in our brains. Proteins are important regulators. They regulate processes such as how our cells divide, how the embryo develops from a single cell to be a creature. Proteins are involved in the immune response. They are all the antibodies are proteins, and we have thousands of different antibodies, each of which fights a different foreign invader. So we have many, many thousands of different proteins, and each of them has a specific function. So the question is, how can we have so many proteins? And here is the answer. So proteins are, if we know how proteins are built. Proteins are built of building blocks, building blocks called amino acids. And here are 4 of these building blocks. We have 20 of different building block amino acids. And in the proteins, these building blocks are joined together like beads on a string. Now, a protein has a length of a couple of hundreds of amino acid building blocks. And the difference between the different proteins is the order in which these 20 building blocks are arranged on these string. So as you can imagine, you can arrange 20 different building blocks in a huge number of different orders or sequences to make different proteins, and that is the answer. How can we have so many proteins? Because we can have different sequences or orders in which the amino acids are arranged in the protein, and these sequences are determined by our genes, by the DNA. So the DNA is the blueprint, and the whole function, or the main function of this DNA, is to give the information for the arrangement of the sequences of the building blocks of the proteins, and therefore each gene, each DNA gene is giving the information for making one certain protein. So this process of joining the proteins together is called protein synthesis. And once the building blocks are joined together, usually the protein folds to make a specific shape, and each protein has a specific shape which is needed for its function, and the shape is determined by the sequence of amino acids in the protein. So when I started out to work in science, there was a lot of interest in this process of protein synthesis, how proteins are made from the building blocks. And there was very little interest in the opposite process of protein degradation. But it was known that proteins are also degraded back to the free building blocks. Now, this looks very wasteful, and maybe because of that, there was very little interest in that. But actually, it was known already at that time that protein degradation has also important functions. And one important function is to to degrade abnormal proteins. So as I said, the protein has a certain structure, and this structure is very important for the function. This structure is very delicate. It can be damaged, and if it is damaged, the protein becomes abnormal and it becomes toxic to the cell. So we don't want toxic proteins, and therefore they have to be removed by being degraded. These abnormal or misfolded proteins are degraded so to prevent toxicity to cells. But there is another important function to protein degradation, which acts on completely normal proteins. We have some normal proteins, regulatory proteins, that have to act only for a very limited period of time. So they are made, they do their action, and then they have to stop their action. And the action is stopped by being degraded. So another important function of protein degradation is to remove normal regulatory proteins to switch off their function. So it was evident that already at that time, to those that were interested, that protein degradation is important, but it was not known how protein degradation is carried out by this high selectivity. And I got interested in protein degradation when I was a postdoctoral fellow in the US. And then I returned to Israel, set up my laboratory at the Technion, and continued to work on the problem of how proteins are degraded. And I continued to work on it because I made one observation in my postdoctoral fellowship, which is that the degradation of proteins in cells needs energy. And that was quite surprising, because the making of protein synthesis was known to require energy. You have to have energy to make these chemical bonds between the building blocks. But it was not known that energy is needed also to degrade the proteins. Actually, it was so that the energy has to be invested to degrade the proteins. So this was the original observation. And when I returned to Israel, I continued to work on it and I asked these questions. How are the cellular proteins degraded in such a highly selective and regulated mode? How does the cell knows to degrade one certain protein out of the thousands of different proteins at a certain time? And the second question was, why is energy needed for degradation of cellular proteins? And I saw that maybe there is a connection between these two questions. Maybe energy is needed for selectivity. Maybe we need energy to make the selectivity, to make the order in the degradation of proteins. So I began to work on it, and we used methods of biochemistry to find out how proteins are degraded. In biochemistry, one opens the cell, one separates the different components of the system that is involved in that system that you are interested in it, and then one puts back together the different components. And if it works again, we understood the system. It is like trying to understand how a mechanical watch works. If you want to know how a mechanical watch works, you open it very gently, very carefully, take out the different parts, the springs, the different wheels, examine them and put it back together. And if we can put it back together and the watch works again, we understood how the watch works. So that is what we did. We tried to isolate the components of the system. It took us several years, but in 1978, we isolated the first component, which was a small protein called ubiquitin. It was called ubiquitin because it's ubiquitous. It's in all cells, but it was not known what it is in its function. So we found out that its function is something to do with protein degradation. It was like finding one or isolating one wheel of that watch, but we didn't know what was the function of that wheel. And here you can see ubiquitin. Here is only the shape of the ubiquitin molecule. It is a small protein. It is built of 76 amino acids. You can't see the amino acids, only the shape. So it's very compact, but it has one end which is free. And we at first we didn't understand why we do need this free end. It turns out that that is the important end of ubiquitin, because here ubiquitin that we found 2 years later gets linked to the proteins that it has to degrade. So ubiquitin is kind of a marker. That's what we found. Ubiquitin is a marker. It marks which protein has to be degraded by being linked to it, by being chemically linked to it, and the chemical linkage is through this free end of it, because that's where it can bind to proteins. So that was the main finding, that the binding of ubiquitin to proteins marks them for degradation. And in the next 10 years or so, we worked on the details of this system, and like trying to find out how the whole watch works. And I won't go into all the details. This is very technical. I just point out that it has two main parts. In the first part, ubiquitin is being linked to the protein. So ubiquitin is represented by these little blue balls, and it is linked to the protein that has to be degraded, which is represented by this orange thing. And in the second part, ubiquitin, the protein which has ubiquitins linked to it, is degraded by this system. And I just want to point out two things. That in the first part, we found that a molecule called ATP is needed for this linkage. Now, ATP is the carrier of energy in cells. So here was the answer to our first question. Why is energy needed for protein degradation? Energy is actually needed to link ubiquitin to the protein, because you are making a chemical bond. And as we said, to make this chemical bond, we need to put in energy. So energy is needed for the linkage. And another thing that I wanted to point out, that several enzymes are needed for this linking, out of which the important ones are the third one, called E3, or the third enzyme. And these are enzymes that recognize to which protein to link the ubiquitin, because that's the important thing, where to link it. Now, enzymes are working like a key in a lock. So each key is specific to a certain lock. And so each enzyme, each protein, each E3 recognizes a certain protein to which to link ubiquitin. And now we know that there are more than 600 different E3 enzymes which link ubiquitin to different proteins, and that is the answer to the second question. What determines the high specificity of protein degradation? How does the cell know to which protein to link ubiquitin? It does it by the selectivity of these different E3 enzymes, and they are the ones that recognize to which protein to link the ubiquitin and then to target it for degradation. So that's the first part of the system. The second part of the system is the degradation itself, and that is carried out by this structure called the proteasome. It is like a barrel to which the protein that has been linked to ubiquitin is stuffed inside and chewed up. So it's like a meat grinder, but a very sophisticated meat grinder. It degrades only proteins that has been linked to ubiquitin, that has been marked by ubiquitin, and then when it degrades them, it chops them up to small pieces and then to amino acids, and also to free ubiquitin, which can be used again now to degrade another protein. So that is in a nutshell how the system works, the basic mechanism of ubiquitin-mediated protein degradation. And when we got to here, I thought it's time to find out how the ubiquitin system carries out some important process in our body. And I got interested in the roles of this system in the cell division, because cell division is an important process in biology and in medicine. When cell division has to be used normally regulated, and if it's not regulated, if it gets out of regulation, we have a disease, we have cancer, because in cancer, cells divide when they should not divide. So it's important to find out what determines the regulation of cell division. And I was interested in the roles of the ubiquitin system in there. And here is what is called the cell division cycle. I took it out of a textbook, biology textbook. So this is a process in which a cell grows, it duplicates its contents, duplicates its DNA, and after the DNA is duplicated, it's divided in a phase called mitosis, or the M phase, into two new cells, each of which has exactly the same DNA as the first cell, and so on. This can be repeated, and we have more and more cells in this process. So that was known for more than 100 years, but was not known what are the molecules that make these cell divisions. And important research by others found out that there are molecules that are called cyclins that act at certain time of the cell division cycle. Here the cycle is represented in a linear fashion. So we have different cyclins that go up in a certain time, a certain phase of the cell division cycle. And then to go to the next phase, it has to be degraded so that the second cyclin will come up that will push the cell to the next cycle. So degradation of cyclins is important, as important as the synthesis. And there are also inhibitors. There are also negative regulators. For example, there is an inhibitor called P27 that inhibits the first cyclin. So as long as the inhibitor is high, the cyclin cannot work. And indeed, in non-dividing cells, in cells that should not divide, the level of P27 is high. And only when there is a signal for cells to divide, then P27 is degraded so that the cyclin can act. So there is a lot of selective protein degradation in the cell cycle, and I ask the question, how is that carried out? What targets a certain protein, a cyclin, or P27 for degradation at the right time? And I thought it might be E3 ubiquitin linking enzyme. Indeed, together with my colleagues, I isolated and studied two important ubiquitin ligating enzymes, or E3s. One is called APCC, that targets for degradation certain cyclins and other proteins that are important to get out of mitosis and to start another cell cycle. And another is this, which targets for degradation P27. So I have a couple of more minutes. That's about my work. And other people got interested in it, in the ubiquitin system for protein degradation. And it turned out that it's very important in health and in disease. Here is some processes in which a healthy cell needs ubiquitin-mediated degradation of regulators, and one is control of cell division. And I already mentioned a couple of examples, and there are many more. In transduction of signals from outside to inside the cells, the ubiquitin system is involved. In the regulation of gene expression, in how a certain DNA is becoming or expressed as a certain protein. It is involved, especially in expression of genes involved in responses to inflammation and immunity, in the development of the embryo, in certain parts of programmed cell death. So in all of these, ubiquitin-mediated degradation is involved in the timely degradation of positive or negative regulators. And of course, ubiquitin-mediated protein degradation is involved in the quality control of proteins, in removing abnormal proteins, as I mentioned. And that's only a very partial list. And it turned out that ubiquitin-mediated degradation of proteins is almost as important, or as important as protein synthesis in the control of the normal cell physiology. And the work of others also showed that this process, the ubiquitin-mediated degradation, is important in diseases. And when the system goes wrong, there are many types of cancer in human, and we'll talk more about that. There are some neurodegenerative diseases, important neurodegenerative diseases such as Parkinson's or Alzheimer's disease. In these, there is a lack of degradation of some abnormal proteins in these cells. As I said, abnormal proteins are usually normally very efficiently degraded and removed. But in these cases, they are not removed in these cells, and these abnormal proteins are toxic, and they contribute to these awful diseases. Ubiquitin-mediated degradation is involved in some types of mental retardation, in the wasting of muscles that happens in some bad diseases. So ubiquitin system is involved in many diseases. And here's just an explanation of how the ubiquitin system is involved in cancer. Cancer, as I said, is due to lack of regulation of cell division. And normally, cell division is regulated by a kind of a balance between the action of proteins that stimulate cell division. These are called oncoproteins. So oncoproteins stimulate cell division, and the opposite process of inhibiting cell division is carried out by proteins called tumor suppressor proteins. So normally there is a balance between them. But if there are too many oncoproteins, there is too high level of oncoproteins, the cell division goes on even if it doesn't have to be, even if there is no, without regulation, or if we lose the tumor suppressor protein, or there's also uncontrolled cell division. So in cancer, there is usually either high levels of oncoproteins or low levels of tumor suppressor proteins, or both. It's like a car. Either the gas pedal is pushed in too much, or we lose the brake pedal, the tumor suppressor. Then the car goes without any control. So what is not known generally is that many oncoproteins and tumor suppressor proteins are turning over. They are synthesized and degraded all the time. And in many cancers, there is lack of degradation of oncoproteins. So the oncoprotein is made, synthesized, but it's not degraded, so the level goes up. And the tumor suppressor protein, many times, it's degraded too rapidly. And therefore we lose the levels of tumor suppressor proteins. And there are many cases in which human cancers are either due to lack of degradation of oncoproteins or too rapid degradation of tumor suppressor protein. So there is a lot of information, a lot of knowledge about the basic system, basic involvement of ubiquitin system in health and also in the diseases. And recently, the pharmaceutical industry got interested in it and tried to develop new drugs that are targeted against the ubiquitin system for protein degradation. And there is already one drug which is very valuable. It is called Velcade or bortezomib. It's an inhibitor of the proteasome, and it inhibits, or it's highly efficient for the treatment of a certain cancer, a bone marrow cancer called multiple myeloma. It's very effective against it, and it allows many years of good quality life for this patient. So, and now it is used for many thousands of patients all around the world. So for me, it's very satisfactory to see the basic science that I have been doing is now being used for the treatment of patients. And we'll hear more about it, and I want to emphasize it for the younger people. Basic science is very important because that's how you begin to get new knowledge. And first, some types of basic science look to you as if just trying to satisfy your curiosity. You indeed satisfy your curiosity, but while you are doing that, you are making discoveries that then later on, usually years later on, will be used for some good application to humans. So I want to conclude by thanking people who worked with me in the Technion laboratory. For many years, I got help from these people, Dvora Ganot, Hannah Heller, Esther Eitan, Sarah Elias, Clara Segal, and my wife, Judy, who is here, and she helped me a lot, also in the lab. And I have many graduate students. Among them, Aaron Chachanover was my graduate student at the beginning of the discovery of the role of ubiquitin in protein degradation. And because of that, he shared the Nobel Prize with me. And also, I had important collaboration from Erwin Rose from Fox Chase Cancer Center, with whom I was also very glad to share the Nobel Prize. And here are people who are working with me now: Daniel Sitri, Shirley Miniovic Shemtov, Sharon Kesaria, Dart Leichner, and Tina Schomer. Thank you very much for your attention.
Thank you very much, Avram. So, young people, do you have questions? You should have questions. Who starts?
Okay, you may ask me later. Or if you.
So? Okay.
About the 24-hour cycle, is that just a round number, or is that how accurate is that number, the cycle?
The cell cycle. The cell cycle is actually not 24 hours. It's different times for different cells, for different organisms. So some cells are dividing in 18 hours. Some cells, it takes them 3 or 4 days to divide. So it is determined. It is not the circadian clock, which is determined by the light and darkness. It is determined by the rate of the synthesis of DNA, actually. That is the most important determinant. When the DNA is duplicated, when we make double amounts of the same DNA, so after that is called replication. When the replication is done, there is a certain time after that, and then the cell division starts, the mitosis. Yes.
Okay.
Thank you. I assume ubiquitin is a protein, correct?
Yes, it is a protein.
So who degrades the ubiquitin?
Well, ubiquitin is degraded very slowly. The half-life time of ubiquitin is about 10 hours. So in 10 hours, about 50% of the ubiquitin molecules are degraded, which is called quite slow. For example, cyclins that are mentioned in the cell cycle, they are degraded within minutes, within 2-3 minutes. And the thought is that ubiquitin, actually, it is very interesting why it is not degraded sooner, because it brings the protein to the proteasome. So it is right next to the mouth of the lion, if you want. And the lion chews up the protein without chewing up the ubiquitin. The ubiquitin escapes; it remains outside. So one of the thoughts is that nothing is perfect. Nothing is perfect in life, for example, for sure not. So maybe it is the imperfection of the system that some molecules are chewed up nevertheless by the proteasome, some molecules of ubiquitin.
Yes? What's for you? Yeah?
Thank you. At the beginning of your talk, you say that abnormal proteins are degraded for lack of toxicity.
Yes.
And at the end of your talk, you say abnormal proteins are not always degraded and then become toxic and can lead to cancer. Can you explain why?
Yeah. Well, as I said, nothing is perfect here. And these diseases, so ubiquitin system degrades abnormal proteins very quickly. Yeah. But it does, for some reason, it doesn't degrade them as efficiently as usual in these diseases. Now, these diseases, the neurodegenerative diseases like Alzheimer's disease or Parkinson's disease, they develop very, very slowly. It takes years until usually and until they develop. So the abnormal proteins are also accumulating very slowly. Now imagine that you have a process which is normally 100% efficient, and here in these cells it's only 99.99% efficient. Still, in years, these abnormal proteins will accumulate, and then they will do it. So our challenge is to find out how we can make the system more perfect or more active in these cells. Yeah.
Okay, one more question. Yes? Ah, you have question? Ah, okay. So stop.
Okay. Preference to younger question.
I didn't quite understand from what the meat grind of proteins are made of. I saw under the picture, I don't know, 268, but I don't understand the word next to it.
260.
The meat.
Okay. The meat grinder, so-called meat
Yeah,
grinder,
yeah.
the proteasome. The proteasome is it's much smarter than a meat grinder, because meat grinder will start and will grind up anything. Here, the proteasome can distinguish between a protein that has ubiquitin linked to it and to proteins that don't have ubiquitin linked to it. So it makes the selection, and it does it by some receptors that it has on it, receptors for ubiquitin. So it is also made of proteins and everything. So some of the proteins of the proteasome are receptors that recognize ubiquitin, and that's why it will push inside only proteins that have been linked to ubiquitin. And then inside it has the grinding, actually. It has the enzymes that chew up proteins. So any protein that gets inside is chewed up. But not every protein gets inside. Only proteins that has been linked to ubiquitin, because the proteasome recognizes these proteins by their receptors.
Thanks.
One more questions? Okay. So I apologize for you.
Okay.
Well, in class, when we talked about your work, we mentioned that the ubiquitous, he recognized what protein is deformed by the MHC, correct? MHC, it's like maybe I'm saying the wrong letters. It might be this, but well, how exactly does the ubiquitous find
which protein to link to?
Yeah.
So it's not the ubiquitin; it is the ubiquitin linking enzyme,
Yes.
the E3s that I talked about. So there are enzymes whose action is to recognize proteins that have to link to the ubiquitin. So these E3 enzymes, on one sense, can carry ubiquitin, but then they carry ubiquitin only to the proteins that have to be degraded. As I said, an enzyme is like a key with a lock. So the enzyme, the key recognizes a certain lock. You can go with your key to your apartment, not to the neighbor's apartment, I hope. So the ubiquitin ligating enzyme, the E3 enzyme, does this job. So so the smart thing is not the ubiquitin. The smart part, or the main part. Everything is smart, of course, in nature, but the main part, or the recognition part, is done by the enzymes, by the ubiquitin linking enzymes, the E3s that recognize the proteins. And as I said, we have 600 different E3 enzymes. Of course, we have many more proteins. We have 20,000 proteins, out of which thousands are degraded. So one E3 usually can recognize a whole group of proteins that have a certain signature.
Okay, thank you very much, Avram.